Good morning, good afternoon, and good evening to everyone joining us from wherever you are around the world. Distinguished guests, ladies and gentlemen, it is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with our main presentation, allow me to provide a brief overview of the initiative that brings us together today. INSTAL Africa, Innovation, Science and Technology for Action and Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. INSTAL Africa was born from a collaboration between the Foresight Institute of Research and Translation Rwanda, or FIRAT Rwanda, and its technical partner, Drosophila UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, it is now my distinct pleasure to introduce our invited guest speaker.
We are honored to introduce our distinguished guest speaker, Professor Ruben Artero, PhD. Professor Artero is a full professor of genetics at the University of Valencia and a principal investigator at the Translational Genomics Laboratory at INSTAL and Siberia. His institutional leadership includes serving as full professor of genetics at the University of Valencia since 2009. He is a principal investigator at the INSTAL Translational Genomics Laboratory and leads rare disease research efforts with Siberia. Professor Artero's international academic journey started with a PhD in biochemistry from the University of Valencia. This was followed by a 6-year postdoc at Memorial Sloan Kettering Cancer Center, New York. He returned to Spain in 2002 with the prestigious Ramon and Cajal contract and attained full professor and principal investigator status in 2009. His core expertise lies in scientific leadership and research, specifically advancing the frontiers of translational medicine for rare genetic disorders through multidisciplinary innovation. His focus includes myotonic dystrophy, where he works on antagomere-based therapies, spinal muscular atrophy using Drosophila and mouse models, and pioneering innovation for limb girdle muscular dystrophy, LGMDD2. The Translational Genomics Lab operates from bench to bedside, researching diverse preclinical models. The lab specializes in drug repurposing, oligonucleotide-based therapies, and innovative in vitro microtissues. Key research methodologies include model systems like Drosophila, mouse, and patient cells to uncover pathophysiology. Therapeutic modalities involve oligonucleotides and antagomeres to restore protein function. Innovation includes in vitro microtissues and omics for precision disease modeling, and strategic drug repurposing accelerates clinical entry. Professor Artero has significantly contributed to academic impact, resulting in 88 PubMed articles, 18 doctoral theses, and 20 patents invented. He has built extensive collaborations to push the boundaries of genetic science. He is the co-founder of ARTHEX Biotech, a high-impact spin-off company advancing an anti-MIR-based oligonucleotide therapy for myotonic dystrophy into clinical trials. This entrepreneurial innovation translates curiosity-driven science into real human health impact. His key global contributions include restoring function via antagomere-based therapies. He also pioneers Drosophila models for myotonic dystrophy, SMA, and LGMDD2. Additionally, he established the Marie Sklodowska-Curie doctoral network on oligonucleotide-based therapies, bridging fundamental genetics and clinical development. Professor Ruben Artero, we are honored to have you as our guest speaker. The floor is yours.
Not sure if you are hearing me. Okay, so well, let me share the presentation. Can you confirm that are seeing my screen and hearing anything? Well, I am going to assume that it's working. Well, thank you very much for this really nice and complete introduction and the invitation. It's a real honor to be here with you today. What I'm going to share is a little bit different from the typical scientific talk, so I hope that you will find it interesting and enjoy it. Well, as already mentioned, my group focuses on translational genomics and rare diseases, basically. And well, my talk is going to talk about that quite a lot. Well, we can all agree there is nothing more comforting than knowing a treatment exists when facing a disease. But for those treatments to exist and be ready when we need them, teams across time and place have worked to understand disease, to design drugs, and to test them, always with the hope of reducing suffering. Today, I want to share not only the science, but also the journey that goes from an idea to a drug for human use, a journey that goes from flight development to biomedical research, from molecular insight to therapeutic hypothesis, and from an academic lab to a spin-off company. So let me take you back to where it all began with a flight gene. Well, you already know that gene expression is controlled by enhancer elements. These are DNA sequences that control the expression of nearby genes. Flight geneticists before genome sequences were completely available developed clever way to identify genes by their expression called enhancer trapping. This is basically that you can introduce randomly insert into the genome constructs which carry a minimal promoter and a reporter gene like Z. And then you can detect the expression of nearby genes by beta-lactosidase stainings. Using this approach, fly line caught our attention because showed clear expression in the developing photoreceptors, shown here in this green signal in imaginal discs, and also in the central nervous system and in the somatic muscles in the embryo, as shown pointed by these arrows. Well, my thesis was the characterization of the gene detected this way. And as I was saying, because the Drosophila genome was not yet sequenced, we had to clone it the old-fashioned way, and also we defined the main transcripts generated by this transcript unit. And we also made imprecise excisions from existing transposable elements, shown here by triangles, and generated a number of small deletions that were used to investigate the function of this gene in adult tissues, in particular in the Drosophila eye. This required generating what's called mitotic clones, where you create patches of mutant cells in an otherwise normal tissue. In tangential sections through the Drosophila eye, like the one shown here, normal photoreceptors contain structures called rhabdomeres that are seen here as these dark blue circles. In these structures, rhodopsins, which are the proteins that can capture light, get accumulated. Therefore, in the mutant tissue where we didn't detect any rhabdomeres, we can assume that this mutant tissue was basically blind to capturing light. In the muscles, we did a similar characterization, and at the ultrastructural level, as shown here, these are electron microscopy images, we detected that the Z disks were abnormal. See this diffuse electron-dense structures compared to the very tight and very electron-dense structures in the wild type. The disks are the places where the thin filaments or actin filaments get anchored and that limit the sarcomeres, as indicated here, as shown to the left in this diagram. So this also pointed to a problem in the terminal differentiation of the muscles. And the problem in the eye and in the muscles suggested the name for the gene muscle blind. Up to here, this was basically a rather conventional cloning of a developmental gene at the time. But well, science usually takes unexpected turns. And a few years later, in the year 2000, a professor in the University of Florida, Maurice Swanson, found out that these proteins, the human muscle blind-like proteins, abbreviated MBNL, of which there are two paralogs, MBNL1 and MBNL2, with partially redundant patterns of expression and also functions, had the property of binding RNA molecules carrying longer stretches of CUG repeats, as shown in this plot here. As you can see, short number of CUG repeats up to about 50, here shown 11, 20, 35, bound these proteins quite weakly because the intensity here is weak. But above 50, and particularly 74, 97, as shown here, these RNAs were strongly bound by MBNL proteins, as indicated here by the strong signal that you can see in this plot. And we will soon see why this is important, because this links the MBNL proteins to a human rare disease called myotonic dystrophy. What is myotonic dystrophy? Well, a typical patient is illustrated here in this image with characteristic symptoms like muscle weakness, that usually forces the patients to carry different aids in order to walk, for example, a cane, as shown here. And also is characteristic of the disease, the myotonia. This is the delayed relaxation of the muscle after contraction. So, for example, when patients make a fist, it may take up to 5, 6, 7, 8 seconds for the patient to open the hand again. Also, is characteristic reduction in arrhythmias in the heart. These are malignant arrhythmias, cognitive, and also reduction in the expectancy. Well, the disease is caused by an abnormal expansion of CTG repeats in an encoding exon in the DMPK gene, which, when transcribed into an RNA molecule, gives rise to longer stretches of CUG repeats. And these RNAs behave as toxic RNAs because they fold into hairpin structures like the one shown here. And these structures have a strong affinity by the MBNL proteins, specifically indicated here, MBNL1, but MBNL2 can also bind very similarly. And these protein RNA aggregates can be detected in patient myoblast as these dots in the nucleus that confirm that this aberrant molecular binding is taking place. This is confirmed by the data that I showed you previously in the previous slide. What MBNL proteins normally do? Well, MBNL proteins are regulators of alternative splicing of exons. And this can be approximated to what happens when we combine different letters in order to make words. For example, we can make the word ship or sip if we skip the H. In the cell, proteins that control this process usually bind to nearby sequences in these alternatively used exons, for example, MBNL1, in the here by blue icon. Drosophila, during fetal illustrates one of the explanations for main symptoms in the disease. During fetal development, this exon channel messenger RNA gets specifically included, and because this exon contains an stop codon, this triggers the nonsense-mediated decay pathway, and basically these messenger RNAs get degraded, and no protein is produced in during the fetal development. In adult tissue, in adult muscle tissue, this exon 7A is specifically skipped and removed from the transcripts because of MBNL proteins that get strongly expressed in adult tissue. And this stabilizes the time that these RNAs can stay in the cell, gives time for them to be translated into proteins, and provides chloride channel protein enough for its function in adult tissue. In myotonic dystrophy, basically this switch to an adult pattern of splicing fails, and therefore the fetal pattern of splicing is maintained. In this case, limiting the amount of chloride channel protein that is produced in the patients, and therefore explaining one of the main symptoms of the disease, which is the myotonia. In fact, several other muscle transcripts behave similarly and maintain a fetal pattern of exon use, producing proteins that are inadequate for adults, explaining the wide range of pathological symptoms. Well, in terms of therapeutic approaches, we could have explored going after the toxic RNA, the toxic DMPK transcripts carrying the CUG repeats. But because MBNL proteins can be produced from MBNL genes, which are perfectly functional in the disease. The problem, I remind you, is the sequestration of the proteins that produce these genes. We thought that we could approach this disease from a different angle. If we could detect microRNAs, which are small regulatory molecules that recognize messenger RNAs by sequence complementarity as part of the RISC complex, we could repress these microRNAs by the molecule, and therefore de-repress the expression of, and therefore boost the expression of the protein and compensate for the sequestration. Following this approach, after testing about 90 candidate microRNAs, we found that MIR23B and 218 were natural repressors of MBNL1 and 2 production, and as shown here, these are results from HeLa cells transfected with these 2 microRNAs. You can see that the levels of MBNL2 protein are strongly reduced, and the same happens with MBNL1, shown just here, MBNL2 for illustrative purposes. So what happens if we follow this approach of inhibiting these microRNAs in patient-derived cells? Well, in this case, we used molecules called anti-mirs because these are synthetic oligonucleotides perfectly complementary to the target microRNA that carry different chemical modifications. For example, the ones indicated here. We're not going to enter into this level of detail because these modifications improve the stability and the binding of these anti-mirs to their targets in biological systems. We tested these anti-mirs in cell-derived cells, and as you can see here, these are DM1 cells. If we detect MBNL1 in the green channel, you can see here these small dots. This corresponds to the ribonuclear foci that I was mentioning before, and you can see that the levels of expression are basically low compared to a healthy cell. Upon transfecting anti-MIR23B, you can see that the amount of ribonuclear foci went down, and the signal was stronger, both in the nucleus and in the cytoplasm, and something similar happened with anti-MIR218, as shown here in these micrographs. Most importantly, not only the looks, the pattern of expression and the subcellular localization of the protein improved, but also the function of the MBNL proteins as alternative splicing regulators. This can be seen here. Well, the upper band corresponds to the inclusion of the alternative exon, and the lower band to the skipping. You can see here that, for example, exon 11 in BIN1 transcripts in DM1 cells, it's preferentially skipped, but upon the treatment, it gets retained again, which is similar to the healthy pattern. And something similar happens with these other transcripts of ATP2A1. So this was the first evidence that blocking these microRNAs could rescue the fundamental molecular phenotype of DM1. Well, up to this point, everything looked like a standard research story. You identify a potential way to address a disease. You test it in cell and animal models. We also did it in a mouse model of myotonic dystrophy. I'm not going to enter into that detail either. And then you basically published it, and you may think that, well, that's my job. I discovered something interesting, but I'm going to a different scientific question, and someone will take it from here to develop a treatment. Well, in our case, we wanted to make a real difference in patients' life, and we questioned, can we go further, not to just show a proof of concept experiment, but to move it forward towards the clinical use. The big challenge, however, was that the science only gives you an idea, but turning it into a therapy is a whole new mountain to climb. Why? Well, because a drug candidate must fulfill a big and intimidating amount of requirements. First of all, it must be very specific and must show low toxicity, must be very stable, both chemically and in biological systems, must show a favorable pharmacological properties, in particular must target the appropriate tissues for the pathology, in this case, the muscle tissue, must show consistent efficacy in animal models, must show a strong commercial viability and regulatory potential, and clear and protectable intellectual property. Well, in general, because the molecules identified in academic laboratories are far from drug leads, and because there is no evidence of effectiveness in humans, the resources needed to progress from a proof of concept experiment to an actual drug are enormous. And this creates what is often called the valley of death between the academia and the industry. As I was saying before, we wanted to cross this valley of death, and in order to do so, we followed three approaches. The first was aligning research with business needs, and this implied working on adding value to the technology instead of seeking novelty, which is typical for academic research. And crucially, to patent the molecules that we discovered in order for them to be able to progress towards human use. Actually, had we not patented almost 10 years ago, nothing of what I'm going to show you next would have been possible. The second approach was to get training in entrepreneurship. And this was possible thanks to a specific nationwide program and a talented postdoc in the laboratory at the time, Beatrice Lemussi, who received the specific training on how to start and manage a company. And finally, to secure private investment, in this case, Beatrice also raised the first round of private funding, which led to the creation of Artex Biotech. That goes from, well, it's an abbreviation from Antisense RNA Therapeutics. And the net result was that we could move beyond the academic laboratory and push the therapy forward towards human use in collaboration with the spin-off company. So basically, we had to thrust in order to move this therapy towards the clinics. Well, the most pressing issue at this point was to move from an academic discovery to a real drug candidate. In practice, this meant designing a large collection of variants, shown here just about 20 of a bigger number of around 100 chemical variants of these molecules, shown here the initial hit, and in different colors, different modifications, different chemical modifications that were known to provide desirable properties to these molecules as human drugs. The next issue, the next challenge was how to classify, how to rank these molecules for the most appropriate, or the ones that had more potential for human use. And for that, we used a parameter called a therapeutic index, which is the ratio between the median toxic concentration and the median concentration, in which you put the two main characteristics that the drug must have, like very potent in terms of efficacy, but must show very low toxicity. Therefore, the higher the therapeutic index, in this case, the wider the separation between activity and toxicity. And using this, and after an extensive screening, we identified the particular candidate, here named improved candidate, that compared to the initial hit required lower concentrations in order to increase the expression of MBNL1 and required far higher concentrations in order to limit or to cause a reduction in 50% in cell viability. Therefore, both things were clearly good for our purposes. After selecting this improved candidate, we expected that the greater potential in vitro would translate into stronger effects in vivo. And when we tested this molecule in the mouse model of the disease, the results were surprising. Using gastrocnemius, we found that even at 100 milligrams per kilo, this improved candidate increased amounts of MBNL1 2.5-fold, whereas the initial hit at 12.5 milligrams per kilo enhanced by 2.3. Also, in terms of myotonia, we saw that the reduction was similar. These mice had 4, which is the maximum myotonia, whereas in both the hit and the improved candidate, the reduction was to 2 in this scale. Therefore, they behave very similar. This lack of correlation between the in vitro and the in vivo activity clearly underscored the need for subsequent optimization of the initial hit in order to enhance the therapeutic potential. To address this issue, we knew that condition to lipids could help because it was reported previously that this could improve the distribution of these molecules. So we designed a second in vitro screening, this time with conjugated versions of the initial antimer, in which in the 5' end of the molecule, we tried several lipid residues, such as oleic acid, palmitic, linoleic acid, etc. And using the therapeutic index, again, we identified a conjugate candidate that, again, worked, increased the amounts of MBNL1 at lower concentrations than the initial hit and required higher concentrations in order to affect cell viability, and therefore was a better candidate than the initial. So by combining the best chemical modifications from the initial screening and the best conjugation from the second one, we ended up with 2 final candidates here named as 107 and 108 with the conjugation. And in order to confirm the contribution of the conjugation to the activity, we compared their activity in vivo with the unconjugated versions shown here without this icon that indicates the conjugation. We tested all 4 candidates, conjugated and unconjugated forms, and the 2 final candidates in the mouse model of myotonic dystrophy at 3 milligrams per kilo, and 5 days later we collected the tissues. Well, the data generated were represented in these radar plots in which each axis corresponds to a different disease parameter. For example, reduction in the microRNA target levels, levels of MBNL protein, different parameters. This is the splicing, inclusion of exon 5 in MBNL1 transcripts, etc. Here, the inner hexagon corresponds to values for the myotonic dystrophy mouse, and the outer hexagon corresponds to the values for the wild-type mice. So the values for each candidate in each axis are plotted in between. Therefore, the area of the polygon basically gives you a sort of combined assessment of the level of the rescue. As you can see here, the lighter areas correspond to the unconjugated candidates, whereas the darker shade areas correspond to the conjugated versions. And in both cases, both 107 and 108, the conjugation improves the rescue because all parameters get closer to the wild-type mice, and therefore they are able to rescue more phenotypes than the unconjugated forms. And here I'd like to highlight again that this was at 3 milligrams per kilo compared to the 12.5 or the 100 milligrams per kilo that I showed you in a previous slide. Well, how do these antimers work? Well, at first, initially, obviously, they bind the microRNA and antagonize their activity. But when we looked more closely in this matter, we discovered that it was a little bit more complex. Unexpectedly, we found that the two microRNAs we were working with, 23B and 218, were actually overexpressed in the disease condition. And this this clearly suggested that there was an active repression on the production of the MBNL proteins, in addition to the sequestration that I already showed you at the beginning. And also, unexpectedly, we found that the levels of these two microRNAs also had an impact on the levels of the DMPK transcripts and therefore on the sequestration. So the antimers we propose that have a twofold activity. First, they release the brake on the production of the MBNL proteins and therefore allow more protein to be produced. And because of the reduction in the DMPK transcript levels, they end sequestration, and both activities GIs in order to improve on the molecular and cellular functions that are impaired in DM1. Well, with this, we could think that this was sufficient to propose human use, but as you know, regulatory agencies require a lot of safety data and activity data, and this has to be provided in a specific way, and it becomes even a third complete challenge to address in order to be able to test a drug in patients. In order to do so, one of the first questions that you have to answer is the design of the clinical trial. This may also seem trivial. You want to test the drug in patients, but the problem is what exactly to measure in patients, because sometimes it may not be trivial, and this risks identifying, detecting, or not measurable activity, measurable relevant activity in the patients. Also, even before this point, you must estimate a safe human dose from toxicology studies from animals, which is a task far from trivial. Then you also have to follow good laboratory practice in order for the regulators, the regulatory agencies, to trust your data and certify that the compound can move forward. And these and other steps converge in discussions with regulatory agencies in order to apply for an investigational new drug approval, which is the official green light to treat patients for the first time. Well, could all this be made from an academic lab? Well, I think you already get the sense that it's impossible, in particular also because of the amount of funding that is required. In this case, Artex started with a seed funding round of almost 7 million euros in 2019. 4 years later, in a Series B funding, got an investment of 42 million, led by venture partners fund, and also backed by the European Innovation Council. And this amount of funding was with this funding was able to launch the Artemir trial, in which in 2024 the first treated with this molecule, and even Later, in August last year, a Series B extension round of 32 million euros was necessary to keep on going through this clinical trial path. And this is one of the messages as well. Even though it may seem like a lot of funding already, every time you fulfill one step, in order to move to the next one in this path towards clinical use, you need to raise even more funding in order to keep on going on the clinical on the development pathway of your drug. Well, all of this could not have been possible without the people. Let me highlight specifically here Dr. Estefania Ferreros, who discovered the 23B and 218 microRNAs as repressors of MBNL1 and BNL2, and was pivotal in the translational path of of the anti-MIR molecules towards clinical use. And also Beatriz Diamusi, who co-founded the company at the beginning, and she's now chief scientific officer in the company. Also, I'd like to highlight the funding by agencies, research agencies here in Spain that were critical for the earlier steps in the development of this. And I said in this presentation, be available in bioarchives, but soon hopefully available online. And let me finish with 4 take-home messages. The first one is that rather than intrinsically good or bad translational projects, it's your vision and above all your evidence that moves it forward, because, well, countless number of times things may be complicated and apparently impossible to move forward, but you always can find a way to do so and to keep on moving towards your final goal. If your therapy needs private investment, patenting is not optional, but it's essential. Collaboration between public and private, and also between basic and clinical research, is the way forward. And also consider departing from the mainstream approaches, because you may find your niche. In our case, I think that if we had followed any approach targeted to the toxic RNA, we might have been outcompeted by other laboratories. So I think that following a different path gave us a competitive advantage that in the end was critical. And let me finish with some inspirational words by the Spanish Nobel Prize Santiago Ramón y Cajal, who more than a century ago said that to understand the conditions under which a phenomenon arises enables us to reproduce it or to suppress it as we wish, and makes us masters of it, harnessing it for the benefit of human life. And that vision, the transforming knowledge to benefit for benefit of human life, is what has been driven our journey from flies to drugs. And with this, I'd like to thank you for your attention, and I'll be happy to take questions. Thank you.
Thank you very much, Professor Artero, for that insightful and wonderful presentation. We truly appreciate the depth of knowledge you dropped there. From your last slide, you told us that consider departing from the mainstream approaches. You may find your niche.
Mm-hmm.
Yeah, so, when it comes to, when you say departing from the main approaches, as research groups, we have one major problem, and that is funding, accessing the funding. When I see the money the funding you listed there, it's huge money, and getting such investments in research is not really easy. For researchers that are just coming up, and they want to do something unique, they need people that are really interested in seeing where it's going to. Most people want to put their money where they are sure there will be results. What do you think could be just, let's say, one thing that they should face that could help them access very good funding to, that will help them find their niche?
Are you saying for our academic laboratory, how to align best in order to get funding? Or
Yes, in both academic and those in clinical, if you want to do research, what's the best way to go about getting funding? Yeah.
well, I think it depends on your local characteristics. My feeling is that sometimes we get sort of influenced by fashion, and sometimes in other countries, someone may be working on, I don't know, let's say artificial intelligence now, and everyone wants to do artificial intelligence, for example. But in your own specific environment, perhaps other things are just better, and you may contribute more, even if not something very hot or very fashionable. So I think you have to find your strengths and focus on that, instead of following the lead of others. Because when you actually hear about something, that laboratory is already ahead of you. So I think it's better to keep your own lines of research, even if modest or maybe not very hot in terms of for others. But if you are good at that, that's in the long term, it's going to give you the fruit, because you're good at that.
Thank you so much for sharing. At this point, we leave the floor open for more questions. Please, you can put down your questions in the chat box so that our prof here can actually respond to them. And so I'll go on to one on your slide 10. You told us that science gives you idea, but turning it into therapy is a whole new big challenge. Can you hear me, Prof?
Yes, yes, yes. Yes, no, that sentence was just because myself, I sometimes thought, well, once you have an idea of developing a drug, I was not aware of how much you still need to do. It's a completely new, not a completely new project. It's just a complete new universe, let's say, because as an academic researcher, you may think, well, I can approach this disease this way, the other way. In an academic environment, you can have very good results, but for that molecule to be useful in a patient, then you have to do so many things. You have to improve that molecule so much that explains the amount of money that you need. So it's a completely new story. And I don't know others, but I know most scientists, sometimes they are not aware of that. And they think, well, I discovered this, someone will take it and magically will transform this idea into a drug. Well, that's a huge amount of work and more that you need. That's why it's so critical to patent ideas, because otherwise no one will risk the private money in order to move those molecules ahead. Or sometimes I know that you may find exceptions where academics or public funding have developed therapies, but in general, you need to work with the private sector.
Okay. So in your work, what was one major challenge that you faced?
In this case, well, in technically, I can share a very. It's going to surprise you, because one of the most critical steps was how to test all those 100 candidates quickly, because we needed something like, we needed to test each of those 100 compounds at 5 concentrations and 3 times in order to have triplicates. If you do the math, you need something like 1,500 determinations of MBNL1 levels, and also as much for an internal control. And there is no way to do that by simple Western blot. And doing by ELISA test, it was just too expensive. So we came up with an idea of doing quantitative dot blot, basically spot a little bit of your homogenate, and after a number of washes and things, you put your antibody, and you have a signal. It's just a dot that you have more intense or less intense, and that gives you an idea of the concentration of your protein. And that was critical. I mean, hadn't we come up with this idea, I don't think we could have ranked all those oligos quickly enough. So that was a technical, against the clock problem that we had to solve.
That's beautiful. That's why it's good to work in a team. Working with a team gives different opportunities for different ideas and how to tackle different challenges. So we have a few questions in the chat box, and the first one: could you share an example where findings from Drosophila research surprised you or led to an unexpected breakthrough?
Oh, definitely. There is one that it really surprised me. I don't remember the names of the channels, but I remember there was a protein. It's a transportator, I think, transport protein that keeps our lungs sort of dry and having liquid. And the same protein, well, the homologous protein, also keeps the the tracheal system in Drosophila dry as well. So even human or mammal lung and Drosophila tracheal system have absolutely nothing in similarity. I mean, in terms of the shape, the how they derive from the embryo, but they still share the same strategy in order to physiologically be dry, because that's important for the airflow. So, well, I thought that was amazing. There are other examples like that. I mean, something completely different between a mammalian and the fly, and apparently it looks like they are just different systems, but the physiology found the same solution to a problem.
Wow, that's an amazing discovery there. I guess maybe someone can just follow up on that, on why
Yeah.
it's the same or what led to that. And so we go to the next question. And for young researchers and students in Africa who are interested in genetics and drug discovery, what skills or mindset do you think are most important to develop early on?
Well, I think you can do drug discovery in flies. That would be sort of easy and not extremely difficult to do technically. But my advice is to be aware of the dose. This is something I learned recently, after too many years, perhaps. But the dose, sometimes in the laboratory, you get too excited with things that you see, well, this extract, this whatever, works in flies, works in cells. But then when you do the translation into human dose, that's just a huge amount or just too much. So for research that you want to have some potential for human use, be aware always of the dose and try to do the math and how much does it mean in for a person, because sometimes you have to kill projects early on before you invest a lot of time into that. So the dose, I think that's the most critical thing that you have to bear in mind.
Okay, thank you so much. Does Prof, does that mean that sometimes when you start a project and you see that the future is very blur or the future is not bright for it, it's easier to just end it from the early stage before you invest more and then look for another avenue or another project that can work with your idea?
Exactly. Knowing that, you could you can perhaps look for alternatives, or at least you know that you have to do something with what you are working with. If it is a molecule, you know you have to improve that molecule chemically, modify the molecule, or in our case, we knew we had to make those oligonucleotides better. But it allows you to align your research into the right direction before investing a lot of time into something that may not have any future.
Thank you so much for that. And okay, during your presentation, you also talked about antimirror. The introduction of antimirror led to several positive discovery. So, could you tell us the implication for future therapists? Because when you showed us the value of death, it looks like the future is very fine. We have so much ideas. We have so many things we want to do, but the future is going to be a very long time, not like nearest. What's the potential for future therapies?
With antimirrors, do you mean
Yes, with anti-males, with anti-males.
specifically? Well, we hope that these antimirrors will be the first antimirrors to reach clinical use, because other therapeutic oligonucleotides have reached clinical use, but no antimer is currently being used in humans. So this would be the first one, because with the targeting microRNAs, you have one good and one bad thing. The bad thing is that because microRNAs bind to different messenger RNAs, you are acting on a number of things. But also the good thing, sometimes you aim to target a pathway where there are a number of things that you want to change in order to improve in a pathology, and microRNAs allow you to do that. By acting on 23B, we are acting on a number of messenger RNAs that we know are repressed in the M1. So we aim to those messenger RNAs without having to act on each of them at them. So you have good and bad things with the well with the microRNAs. This has affected the how quickly these molecules have reached clinical use. And so we expect these microRNAs to be the first ones to reach clinical use, and after that, usually happens that others may suddenly become much faster, may fulfill all the requirements faster because of the precedent that you have established.
Okay, that's a beautiful and insightful discovery there, and we are hoping that the future is bright for anti-meal and it can go ahead to clinical application. So one last one, Professor, for those who may be new to research and who may be interested in genetics, drug discovery, can you tell us from you, what was that passion? What actually drew you to this particular work?
Well, the patients. I mean, I think we all share that. That's why my second slide was how happy we all are when we are sick and discover that we take this pill and it go away. I mean, we all appreciate how important it is to have medicines. So I always thought, well, it's nice to be in the laboratory and discover things, but if those things do not translate into actual medicines, then, well, in the long term, we all assume that would be important for someone or at some time. But I always thought I always aimed to actually make a difference and myself be well contribute to that process and be able to provide something to the patients. In this case, if that was finally the case, I would be really happy.
Thank you so much, Professor. Ooh, thank you so much for following your passion to go into research. And we are all looking forward to learning more. On behalf of Install Africa, we're very happy to have you today. Uh, oh, apologies. We have one more question that just came in. And okay, the person said, which AI system can one adopt in drug discovery in vivo using Drosophila melanogaster?
Well, I don't really know, but I'm not sure the artificial intelligence system will, well, for some, at some point you may use that, but the problem with in vivo discovery using Drosophila is probably the the phenotype that you can screen, right? I don't think the AI will tell you that, but you probably have to come up with a good phenotype to screen, something that is clearly relevant for in humans, something that you can translate into a human phenotype, and something that is and something that can be moved towards rescue or making it even stronger with molecules. So I think before the artificial intelligence, you need to come up with a good phenotype to screen, and that's be a human brain still only associated with all the no, be congenital actually. Myotonic dystrophy is a very variable disease. It shows very different presentations, different patients, and it can be it can affect newborns and kids, early adults, older adults, old people. So at any time during your life. It depends on the number of repeats. I mean, if you have closer to 50 CTG repeats, then it tends to appear later in life and weaker. But if you have a long number, a large expansion, it tends to appear earlier in life and to be more severe.
Thank you so much, Prof, for those answers. I hope your questions were all responded to. If there's any more question and maybe you remember after now, you can send us a message. You can drop it on the replay on YouTube, and then we can get your response back from Prof. Thank you so much, Prof. On behalf of Install Africa, we are delighted to have you on this platform. Thank you for the fresh ideas, opportunities, and answering all our questions. In this year, 2026 is a new season for learning, and we start the year with a strong research capacity. The person that asked about AI tools that can be used, well, since we can have those that can be used in vivo, on Firas developed a new it's called Snap Genius. is a research tool that can be used by students, researchers. It helps make your research easier. You can actually explore it. You check on our YouTube channel, we'll have the link pasted there, or you go to snapgenius.tech and have access to it. And then you can carry out your research works there. AI is the thing now, but there are some areas that AI have not been able to come up with. Just as we wait for our results, or we also wait for our antimail to get to clinical application, we are also waiting for our AI to be able to be used for more intense research work. So thank you so much, everyone. This is Foresight Institute of Research and Translation. We have several other programs that come up. We have the Nurses Diary, Mental Health Diary. We have the Dental and Medicine Chronicles. We have different sensitization webinars that go on. So you can subscribe to our YouTube channel so that you have more information. You also see our the discoveries, our new research works from different professors and learned researchers far and around the world. So without any more delay or taking much of your time, we say thank you so much, everyone, for joining us. Thank you so much, Prof, for taking your time to join us on today's event. And we'll, we'll like you to subscribe to our channel and reach out to us anytime you have any more questions. Our lines are always open. Your questions will always be attended to. And with that, we come to the end of our program for today. INSTAL Africa is a program that works on research, and every month we have events. So please, if you did not register to have emails sent to your mailbox, you can follow us on FireArt. You can check us out on LinkedIn too. We also have our event posted there. Thank you so much, everyone, and have a lovely day. Bye.
Okay. Bye bye.